just to introduce what I am, and that's a microbial explorer. And so E.O. Wilson is a contemporary American philosopher, and he said, if I could do it all over again and relive my vision in the 21st century, I would be a microbial ecologist. And that's because it is so much fun. There are 500 to 1,000 bacterial species per mammalian gut, so this area is ripe for exploration. So as Spencer said, despite my soil bacterial training and background, it all applies to complex ecosystems and bacterial interactions with each other and with the host environment and other biotic and abiotic factors. Also, viruses are incredibly diverse. There are 10 million virus particles per milliliter of seawater, and we've counted in some of our fecal samples 1 times 10 to the 9th virus-like particles per gram of feces. And so viruses are an extremely important component of ecosystems also. And so what I came to the NADC to do is to do some of this exploring and hypothesis testing in complex environments. And our unit, I'm sorry I'm going fast, but I'm worried that we're going to be running long, so I'm purposely going fast. We are the Food Safety and Enteric Pathogens Research Unit, and our 2 objectives are to reduce foodborne pathogen load. We've got folks working on vaccines for E. coli 0157H7, Salmonella, and Campi. Also, we're interested in developing or testing pre- and probiotics. For example, our postdoc, Uri Levine, studied a commensal bacterium of pigs, Mitsuokella, and he found that it inhibited the growth of Salmonella. And so this is an example of a beneficial microbe that exists in that complex bacterial community of a gut, and how it perhaps could be turned into a product that we could use to manipulate Salmonella. Also, we've got projects doing functional metagenomics, and this is bioprospecting for small molecules that would inhibit foodborne pathogens. And I'm not going to talk about any of that today, but I wanted to introduce you to our work in our unit, and that picture was of people in our unit. I'm going to talk about the research that I do with Thad Stanton and other scientists in the group, and that's studying alternatives for growth-promoting antibiotics. And so in order to define or discover alternatives, first we need to know how antibiotics are functioning, to participate in growth-promoting effects. And so today I'm going to talk not just about the antibiotic effect, but also a collaboration with Sean Beerson studying the effect on the swine microbiota with Salmonella challenge, because I was tasked to describe what's changing in the swine microbiome, so I'm going to tell you everything that's changing in the next 20 minutes. So first, our biological question I'm presenting is what happens to the swine gut microbiota during Salmonella challenge. So as you all probably know, Salmonella is arguably the most important foodborne disease with a pretty high mortality rate, and I believe these data are from the U.S. Specifically to the swine industry, it's important because over 50% of pig farms test positive for Salmonella, and the goal of any strategy is to reduce Salmonella carriage. And so we wanted to assess the swine gut microbiota before and after Salmonella challenge to see if there are bacterial species or anything, if we can pinpoint anything happening in the microbiota that would give us clues to things we could do to modulate that and help decrease Salmonella carriage. So the experimental design, this study was designed, and this portion of it was conducted by Sean Beerson and her team. It had at least 6 piglets in the non-inoculated group and a huge group of 54 piglets, and these are all in our controlled barns down at the NADC. Whoops, this is sensitive. They were inoculated intranasally with Salmonella enterica or typhimurium, and then they collected fecal samples over a 3-week period, first starting just prior to challenge and then up to 21 days after challenge. They enriched for Salmonella and enumerated them, and then retrospectively categorized the pigs as low or high shedders. So when a pig is challenged with Salmonella, they all shed to various degrees, ranging from what can be called low or high shedders. And so they, after categorizing these pigs, they isolated the DNA from 5 low and 5 high shedder pigs, as well as the non-inoculated pigs, and then we performed 16S rRNA gene sequencing. So first a little bit about the classification. This is called an area under the log curve, and this was developed in Chris Tuggle's lab, and the citation for that method is up there. This figure shows the classification of the shedders from Sean's experiment. And so what you can see here is that there are the 5 data points in red were called high shedders, and then the 5 in green were our low shedders. And I forgot to say the x-axis is the days post-inoculation, so you're reading it over time. And then what is a 16S rRNA gene sequence analysis? Well, the 16S rRNA gene is a gene that's ubiquitous in all bacteria, and it has conserved and variable regions, and so it's an excellent tool for phylogenetic analyses of bacteria. And because of these variable regions, we don't even have to sequence the entire greater than 1 kb gene. We can just sequence a small piece of it that includes 2 or 3 of these variable regions. And we can barcode the DNA per pig, and so we can do highly parallel sequencing reactions of the entire microbiota using Roche's 454 genome sequencer. And so shown here is Leanne Hobbs running our Roche 454 machine. And so then we take all the sequence data that are barcoded, and I analyze them in a program called Mother, and this allows you to quality control the sequences and compare them to each other compared to all the other 16S gene sequences in the database, which is incredibly well supported by the community. Lots of information in there. So how do you organize these data? You can organize them first by operational taxonomic units, and this is grouping the sequences just by how similar they are to each other, and it's without regard for nomenclature. So for example, within this you don't say this is E. coli or this is, anything else. You just group them and see how they're related. And then of course, the thing we can wrap our minds around is by analyzing the 16S based on the closest named relative. So this would be by what looks like E. coli, what looks like Clostridium or something like this. So first I'm going to show you data analyzed by operational taxonomic units. And the reason I needed to go through that background is because it's a little bit complicated to think about, but each of these data points is the entire microbial community of one sample, one pig fecal sample at one time point compressed into one data point. And so this is called canonical correspondence analysis, and it's a tool used in ecological studies because it allows you to plot variables, so in this case shedding, time, and litter, along with your data from the 16S analysis, so frequency counts of the different species as they occur in the fecal sample. And so first here I'm showing you day 0. The blue are non-inoculated, the green triangles are the so-called low shedders, and the red squares are the so-called high shedders. And so at day 0, what you can see here is that both the green triangles and the red squares are overlapping with the blue, which are the non-inoculated pigs. But what is important about this slide is that the green and the red are actually completely separate from each other. And there's a p-value to support this by an analysis of similarity or ANOSIM. And so that's important point number 1 to take with you, and I'll reiterate that, is that the community, the microbiota of the low and high shedders are actually significantly different at day 0, so prior to Salmonella challenge. And then I really love this animation. I was so proud of this. And so over time then, so if you look at the same analysis, same data, day 2 data. So day 2, this is 2 days after Salmonella challenge. What you can see is that the high shedder pigs, the microbiota of the high shedder pigs has completely and significantly separated from both the non-inoculated pig microbiota and the low shedder pig microbiota. And so that is significant and interesting and perhaps intuitive, because with the Salmonella challenge you have a variety of factors such as fever and diarrhea and a lot of physiological things going on in the pig that could be causing the shift in the microbiota. And so now the plot has added, so I forgot to mention, but maybe you can see that the most recent data point is in color, and then I've grayed out the past data points so you can see where the community has been. And so by day 7, the high shedders are still significantly different from the other microbial communities of the other types of pigs. And then by day 21, what has happened is actually pretty interesting too. And that is that the microbiota of the low and the high shedders have become more similar to each other than to the microbiota of the non-inoculated pigs. And so we think that suggests some commonality in the microbiota of pigs that have experienced a Salmonella challenge, and it has perhaps altered the normal maturation of the microbiota from what would have been a healthier non-inoculated pig. And so again, to reiterate, the 3 primary conclusions from that canonical correspondence analysis is that at day 0, the community structure of the Wilby, low, and high shedders, their microbiota was significantly different. At day 2, the Salmonella-induced changes caused significant shifts in the microbiota. And then by day 21, the microbiota of pigs that had been challenged with Salmonella was now more similar in community structure than pigs that had not experienced Salmonella. And so who on earth is who's responsible for this? This is the more interesting part, right? Which bacteria are responsible for these changes? So first, I wanted to tell you that the swine microbiota resembles all mammalian gut microbiotas in terms of, or at a higher level of classification. So over 50% of the swine gut microbiota are Bacteroidetes, and then a lot of Firmicutes, and then the other phyla are represented in minority. And this is common among humans and mice and zoo animals, pretty much every microbiota associated with guts. What's unique to swine is that the relative abundance of Prevotella is extremely high. There's been a lot of research done on Prevotella, and I have not read all of it by any means, and I don't know the significance of it. It might just be the way the swine microbiota is. If anybody knows anything special about Prevotella, I would love to hear it. So which bacteria are driving the shifts at day 2? So day 2 was the day that showed a dramatic shift in the bacterial community structure of high shedder pigs. And so at day 2, this is a stacked bar graph showing on the y-axis the average relative abundance of different bacterial genera, and then the x-axis we have non-inoculated pigs is the first stacked bar, the low shedder pigs, and what's plotted is the average of all 5 pigs. And then the high shedder pigs are the final bar. And then you can see the different bacterial genera that we plotted on the graph. And the symbols just mean comparisons that were statistically significant among all of the animals. And so what's notable about this to us, and you can pick your favorite bacterium and look and see what's happening to it, but what was notable to us is that extreme decrease in the abundance of Prevotella. And so Prevotella is the most notorious swine normal member of the swine microflora. And so it's decreasing, and we supported this result in a qPCR analysis. And so we don't exactly know what it means yet, but this is what's happening to the microbial community of high shedder pigs at day 2 post challenge with Salmonella. And so what was actually most important to us, or exciting potentially, was this difference in the microbial community structure at day 0 between the low and the high shedder pigs. Is there a member of that microbial community that is dictating, for example, the lower shedding status? Is there a bacterium that we can target as probiotic or with a prebiotic to influence other pigs to be low shedders too? Disappointingly, there was no single bacterium that was responsible, for the difference. But it was perhaps not surprising because there are it's a very complex microbial community, and there were these were the presented on this dot plot are the 10 bacteria that were differentially present between the low and the high shedders at day 0, and their P was all slightly. It was the lowest P value that I had. Are there trends? you can see just between the difference in the green triangles versus the red squares, you know, they're overlapping, nothing significant. So we don't hang our hat in any of these bacteria. We think it's the there's something about the microbial community structure between the low and the high shedders, and we're doing subsequent experiments to tease that apart some more. Specifically, we're interested in doing a systems biology analysis of Salmonella shedding, and so I've showed you what we've learned about the bacterial membership related to Salmonella shedding. And Sean's collaborator, Chris Tuggle, and co-authors have a lot of swine gene expression data from the exact same pigs, so parallel study. And so our next step now is that we want to see if there's correlations. We're sequencing the fecal microbiome of some additional pigs to try to increase our effect size with the statistics, and those pigs will be matched with the swine gene expression data. So stay tuned for some hopefully exciting results there. How am I doing on time?
A few more minutes.
A few more minutes? All right. So the other half of what I came to talk about was the effect of antibiotics on the swine gut microbiome. And I suppose it's fitting that I'll rush through this because this is all published, so you can access this. The data I just presented are submitted, so those will be available soon. So why, we already know, we've heard a lot in this meeting about the importance of alternatives to antibiotics in agriculture, at least for the non-therapeutic or growth-promoting uses. So the FDA recently published a guidance, and consumers want alternatives, and it's complicated. So what I think is even more complicated are alternatives themselves, and that's because how antibiotics work to promote growth is, I think, unclear. So there's a lot of different things in the literature. Are growth-promoting antibiotics preventing pathogens? Are they anti-inflammatory? Are they decreasing the pressure on the immune system? And we recently submitted a schematic representing this. And so antibiotics, are they somehow decreasing or anti-inflammatory? Are they inhibiting pathogens? Or are they somehow modulating the gut microbiota in a beneficial way? And so for alternatives to be efficacious, we think it's unlikely there's going to be a magic bullet alternative to an antibiotic. It's going to need to be some multifactorial approach, including perhaps anti-inflammatories, prebiotics, and vaccines. And so, again, to get at efficacious and thoughtful alternatives, first we want to know what's the effect of growth-promoting antibiotics on the indigenous microbiota, and I will just skip the phage part. So our big experiment that we conducted while I was a postdoc at the NADC, we had 4 groups of 6 pigs, all in separate rooms in highly controlled environments. And we fed one group, and our control pigs served as controls for the entire duration of the experiment. So we fed some pigs Carbidox at 2 different levels, and the third group of pigs we fed ASP250, which is a blend of penicillin, chlorotetracycline, and sulfamethazine. And so I'm going to talk about some of the ASP250 results first, and I think the Carbidox I won't get to today. So ASP250, we conclude alters bacterial membership. So this is a principal components analysis, and although it's complicated mathematically, it's simple like the previous one in that each data point represents the entire microbial community of a single sample. So one fecal sample, one time point, one treatment. And so it's a way of showing all of your data in 2 dimensions, which humans like to see things in 2 or 3 dimensions. So our circles are the non-medicated pigs, and the squares are our ASP250 treated pigs. The black is day 0, which is were samples taken prior to starting the ASP250 treatment. And then the colors, blue to pink, are after 8 and 14 days of continued ASP250 in the feed. And so what's notable about these data is that all of the colored squares separate from all of the other data points. And that is at a statistically significant, in a statistically significant manner, with an interesting R value. And so what's this showing is that the microbial community structure is in fact being altered in a significant way by the ASP250 in the feed. And so like before, which bacteria are responsible for that shift? We found a couple of different bacteria that are decreasing with the ASP250 in the feed, Coprococcus, Succinovibrio, Trisibacter, etc. And so again, if anybody sees their favorite bacterium, come talk to me. It'd be fun to hear what you know about it. What we thought was most interesting to start with was that E. coli was increasing, and we had seen that in our previous experiment with actually much smaller samples, but in that previous experiment we also saw E. coli populations increasing in medicated pigs. And so that got us thinking, and I should also say that we tested, I think only 100, but we tested a panel of our E. coli isolates in a multiplex PCR assay for pathogenic strains, and they were not pathogenic. So these are just normal commensal E. coli blooming in the guts of pigs that are fed ASP250. So we got to looking in the literature, and actually, I don't even think we started off looking. We just noticed in some other studies with other antibiotics in other animals, chickens and so forth, E. coli was also blooming. So then we started looking, and we found that E. coli bloomed in pregnant women, in starving kids in Bangladesh, with diet changes in cattle. So it's reported, it's kind of sprinkled throughout the literature, this E. coli bloom. And so we hypothesized in an addendum recently in gut microbes that maybe E. coli is an indicator of a general gut disturbance. We recently had someone contact us about E. coli blooming in a colitis model in humans. So we don't have anything we're doing with this yet, but we think there might be something interesting there with this phenomenon of E. coli blooming. Am I done?
Can be.
The other things I was going to talk about were functions of the community, and so I'll just say we have an interest in mucin degraders and butyrate producers. Butyrate is important for colonic epithelial cell health, and so we've identified some bacteria that produce butyrate, and actually graduate student Tori Luft, in collaboration with postdoc Rory Levine, have identified an isolate of Cloaca bacillus that can both degrade mucin and produce butyrate. And so we think this is a really interesting example of the symbiosis that occurs with the microbes in the gut, and we hope to exploit that someday. We also study antibiotic resistance because of its importance to the antibiotic debate. And then I'll just go right down through to the acknowledgments. So I'd like to thank Thad Stanton. He's our research leader and started this project. The support scientists and students and technicians, some of whom I've presented their work. And then also on the Salmonella project, primarily Sean Bierson, but also her collaborators. And I'd like to also thank Jim Teege and Tim Johnson for work I didn't present on the antibiotic resistance work that we did. So.